Hey everybody, in this video I am going to talk about some neat DNA elements called enhancers, and I did not spell that correctly, so enhancers. So enhancers, what are these things? So enhancers are related to transcription, and we've already learned quite a bit about transcription, haven't we? So what have we learned so far? Well, obviously I can't talk about everything that we have learned with respect to transcription, but I just want to mention that we have talked in great deal, or I have talked at great length on transcription initiation and how it depends on the general transcription factors, and they help recruit polymerase II to the promoter region of POL2 transcribed genes. And these alone are thought to be sufficient for initiating transcription. However, alone, transcription is not very efficient. So we have other proteins called transcriptional activators and transcriptional suppressors or repressors. Repressors or suppressors, whichever one you want. I'm going to go with suppressors. I'm sure they're used interchangeably. So what do these do? These both bind to DNA. Activators bind to elements known as enhancers, which is going to be the main topic of today's video. Suppressors bind to DNA, and they tend to bind to elements called silencers. So at some level, an enhancer and a silencer can be the same piece of DNA, same segment of DNA, but it acts as an enhancer when a transcriptional activator is bound to it, or it acts as a silencer when a transcriptional suppressor is bound to it. Essentially, we're going to focus on the enhancers in this video. So, okay, how do enhancers work? Let me talk about them specifically for a moment. So I'm diagramming a locus here, and I'm going to use slightly different variations on this diagram here throughout this video and the next video. So let's say this is an enhancer over here, and let's say this is a gene. We'll call this gene A, and we'll call this one gene B. And let's say this is the promoter region here and here. Nope, that didn't work. I tried to get fancy. Nope. Okay, let's see here. There we go. Okay, so the red rectangles are the promoters. These are the +1 sites where transcription starts. Now, this enhancer right here, let's say this enhancer increases the transcription rates of both of these genes right here, even the one that's located very far away. So that's one of the most interesting things about an enhancer, is they can work on genes that are located very far from the enhancer. So enhancers are generally 50 to 1500 base pairs long. They can influence transcription of genes that are up to 1 million bases away. So probably more, but I'm guessing the 1 million base pairs away is maybe one of the longer distances that has been associated with an enhancer. They can work both upstream and downstream of a transcription start site. They can be found within a gene, within an intron of a gene. And they function independent of orientation. So you can take the enhancer, flip it around at the same spot, and it still works. So what I want to focus on is, how does that work? How does an enhancer have all these different characteristics, and how does it influence the transcription of a gene that's located so far away? And why does the orientation? Why doesn't it matter? So there are all these interesting things about enhancers, which hopefully I'm going to be able to explain to you in this video. So enhancers work through, well, we already said that transcriptional activators bind enhancers, but co-activators are needed for those enhancers to promote transcription of a target gene. So probably the most important co-activator in eukaryotic cells is called the mediator complex. Now, this is a giant complex made up of up to and slightly over 30 subunits, so 30 protein subunits. And the mediator complex is known to interact with general transcription factors, RNA polymerase II, transcriptional activators, and transcriptional repressors. So through all these different interactions, the mediator complex is, thought to mediate interactions between the enhancer and the promoters of target genes. So we have a model of how these enhancers are thought to work at such long distances and why the orientation doesn't matter so much. So here's a slightly different diagram with respect to the diagram I had just produced for you. So same elements, but I'm going to diagram the elements in sort of a different way relative to one another than what I did in the previous diagram. So these little things here, they're just saying that the chromosome goes, off the screen at that point. Now, here's the enhancer that I previously diagrammed. Here's gene A, here's gene B, and the enhancer works on both gene A and gene B. It's easier to see how the enhancer may influence gene A because, it's right next to gene A. But gene B was further away. So what we have is this chromatin looping, or chromatin loop. And through this loop, because the DNA is, bent here, that this enhancer here can be brought in close proximity to the promoter of gene B. So here's the +1 site there, here's the +1 site there. Now, these little red circles, these are the transcriptional activators bound to the enhancer. This green thing right here, this is going to be the mediator complex. And I haven't diagrammed some other things here, right? I haven't put the GTF, so let me put this yellow thing here. This is going to represent the general transcription factors, like all those general transcription factors that associate with the promoter. And okay, so now we have this model of how the enhancer can influence transcription of gene B when it's so far away through a chromosome loop, so our chromatin loop. Now here I could put all the various nucleosomes. I'm not wrapping the DNA around the nucleosomes, but I'm just showing nucleosomes here. And this loop can be held in place by a complex called cohesin, which we didn't talk much about in this course. So that's called the cohesin complex. So here we have our model. Now, in recent years, researchers have found that enhancers are transcribed by RNA polymerase II to make enhancer RNAs. So there's an RNA polymerase II, and if we have RNA polymerase II loaded on DNA and transcribing RNA, well, we can probably bet that the general transcription factors are going to be there also. And okay, they are transcribing RNA using the enhancer as the template, and that RNA is going to be called an enhancer RNA. Okay, so we have these enhancer RNAs. We have enhancers. Now, our next question is, are these enhancer RNAs necessary for the enhancer to function properly? So we know the enhancer is able to promote or help promote transcription of target genes. In this case, the target gene is gene B. Are the enhancer RNAs important for function? And that's what we're going to take a look at in the next part of this video. So, okay, so this is our model of how the enhancers can work to promote transcription of a target gene that is located at far away from the enhancer. And you can see that if we flip this enhancer around, well, you know, it's still located. You know, the chromatin loop can still form. It's still located near the promoter of the target gene, so that makes sense that it could work no matter what the orientation is. So we're starting to see how, you know, these enhancers can work regardless of the orientation of the enhancer and regardless of the far distance from the target gene. Okay, so we have these enhancer RNAs, which are a relatively recent discovery. And what I want to do is look at a paper from Xie, and I hope that's close to the correct pronunciation of that name, Xie, et al. And this is in 2014. And we've learned a little bit more about enhancer RNA since then. But I thought this was a pretty good paper, and I want to talk a little bit about what they discovered with respect to enhancers and enhancer RNAs. So they are looking at an enhancer, or they looked at an enhancer called KLK3E. Now, the KLK3 enhancer has a couple of target genes. And I diagrammed that a little further away than I wanted to, so let me back up here. So one target gene is called KLK3, and another one, which is located much further away, is KLK2. And these are human genes. This is a human enhancer, and it may be conserved in other mammals. I'm not sure. So just some basic studies on the enhancer. How do we know it functions as an enhancer? Okay, so one of the things they did was they created 4 different plasmids. Now, they did a lot in this paper. I'm just going to focus on a few of the experiments that they performed. So we have 4 different plasmids here. And so each of the plasmids is going to have a promoter. And in this case, it's going to be the KLK2 promoter. So the promoter for the KLK2 gene. So KLK2. Now, instead of the KLK2 gene, they are going to use the coding sequence for a protein called luciferase. So when these plasmids are placed into a mammalian cell, RNA polymerase II will bind to the KLK2 promoter and transcribe a messenger RNA that encodes the luciferase enzyme. So an enzyme called luciferase. Now, okay, what is luciferase? So just briefly, well, luciferase is an enzyme So luciferase is an enzyme that acts on luciferin and uses ATP to make oxyluciferin plus light. So it releases a photon of light. A photon of light, is that redundant? Releases a photon. Plus AMP. So luciferase can make, it's a chemiluminescent reaction where it processes luciferin into oxyluciferin and releases a photon in the process. And so why do molecular biologists use luciferase in experiments like this? Well, it's really easy to quantify, how much luciferase enzyme a cell produces if you have this luciferin substrate that you can add to the cells, and you have a machine that records the amount of the number of photons that are produced in a reaction. So, okay, so all of these orange bars, those are the coding regions for the luciferase enzyme. And we can say the +1 site is right here. It's the same for each. Now, they also took the KLK3 enhancer and put it in the plasmid upstream of the KLK2 gene, promoter, sorry. Upstream of the KLK2 promoter. And KLK3, so in one orientation, they checked it in the other orientation, and then they had one with no KLK3 enhancer, and they also had a plasmid, I should take this out, a plasmid with no enhancer and no luciferase. No luciferase coding sequence. So they take these plasmids and they put them in a mammalian cell line. So the cell line isn't that important. And, I looked through the paper and it wasn't actually clear with this experiment what cell line they used, but it could be mammalian cell line. It may be a cancer cell line called LNCAP, which is a prostate cancer cell line. It doesn't really matter. It is a cell line where the enhancer can work if it is present. So they have those various plasmids. And okay, so what did they do? They transfected, which essentially means to put the plasmid into the mammalian cell line. And then they checked each cell line with the different plasmids for luciferase activity. And the more luciferase activity, the more transcription of the luciferase coding region on that plasmid. So let's just say, take the control first. So no luciferase coding region on the plasmid. So that's just a control. We're going to have a value of 0 for that cell line with the plasmid that doesn't have the luciferase coding region. So what about the plasmid that has the luciferase gene attached to the KLK2 promoter, but no enhancer? So that one, we're going to get a slight, we're going to have a sort of basal level of luciferase activity. And these levels here, these are all going to be relative. It might be a log 2 scale. That's not really important. What's important is the height of these bars here. So the taller the bars, the more luciferase activity we have. So which ones did we look at? We looked at this one. Okay, no luciferase activity. We looked at this one here with no enhancer, and we saw sort of a basal level activity, saying RNA polymerase 2 is binding here at a certain basal level and transcribing the luciferase gene. That's being translated to give us the luciferase enzyme, and the amount of enzyme is allowing us to... Someone just knocked on my door. Hold on. Okay, I'll keep going. The amount of luciferase activity is allowing us to estimate the amount of enzyme in the cell. Okay. And the amount of enzyme in the cell is related to the amount of transcription. Okay, so what's next? So the next one... Let's take a look at the plasmid with the KLK3 enhancer, let's say in the forward orientation. Forward orientation, reverse orientation, doesn't really matter. Someone's still at my door. I'm going to check that and be back in a second. Okay, sorry about that. I had a visitor at my door who needed information. Okay, so now we're taking a look at the plasmid with the KLK3 enhancer in the forward orientation. And what we're seeing is, whoa, ton of luciferase activity, saying that luciferase gene is being transcribed at much higher levels than in the plasmid with no enhancer. So the enhancer is working like we expected it to. Now, the same thing happens when KLK3 is in the reverse orientation. And if you look at the paper, what you'll see is that, I have these bars at exactly the same height, but they're slightly different, and they do some statistics with replicates, and they have error bars, and the error bars are essentially overlapping. So there's no significant difference between the plasmids that have KLK3E in the different orientations. Now, interestingly, this is sort of a new finding, researchers have known for a long time that, they could probably predict you take an enhancer and it acts on the promoter that's in the plasmid, you're going to see more transcription. So they also looked at, just looking at the forward orientation now, and we can predict that whatever we see for the forward orientation, we're probably going to see for the reverse orientation too. But we're just going to look at the forward orientation. Now they added, with the plasmid that has KLK3E, the KLK3 enhancer in the forward orientation, they added KLK3E specific siRNAs. And some of you might remember from 219 genetics what siRNAs do. We're going to review those in an upcoming lecture. But for now, just know that eukaryotic cells have a mechanism called RNAi for RNA interference. So RNA interference, there are a number of proteins, or several proteins involved in this mechanism. A few of these proteins form a complex called the RNA-induced silencing complex, or RISC for short. Now, what RISC does, it binds to these short interfering RNAs. In the short interfering RNAs, they start as double-stranded often, but in the RISC complex, they are single-stranded. So single-stranded, 21 to 24 nucleotides long. And what the RISC complex does is it uses the siRNA, which is for short interfering RNA, to scan RNAs in the cell. And if an RNA, like a messenger RNA, for example, or an enhancer RNA, has a segment that is complementary to the sequences in the siRNA, what the RISC complex will do is it will cleave the RNA that matches the siRNA. So that RNA is no longer functional, can no longer be translated into a protein. If it is a messenger RNA, or if it's an enhancer RNA, it may not function. Probably won't function if it's, cleaved into two pieces. And then once it's cleaved, it is usually degraded by fully degraded by other exonucleases, or nucleases. And there are other processes by which, instead of cleaving the RNA, the RISC complex can just bind to it and prevent it from doing whatever it's supposed to do. So that's just a brief overview of what RNA interference does. And we'll talk more about that in an upcoming lecture. So what they found, however, when they added siRNAs with the plasmid, and the siRNAs are complementary to the RNA made by the enhancer RNA from the KLK3 enhancer. When they're complementary to that, we get much less transcription of the luciferase gene in the plasmid. So let me back up here. We're using this plasmid with the KLK2 promoter attached to the luciferase gene. And this is the KLK3 enhancer. So when this plasmid is placed into a cell with siRNAs that match the enhancer, Well, the enhancer doesn't work that well, and we don't get much transcription of this luciferase. Their control experiment is the KLK3 enhancer plasmid with the enhancer in the forward direction, plus control siRNAs. So usually the control siRNAs have the same nucleotides that are in, let's say, the KLK3E siRNAs, but they're shuffled. So the nucleotides are the same, but they're not in an order that matches anything. So they're not complementary to the enhancer RNA from KLK3E, but they have the same nucleotides in a useless order. And when they use these, okay, they still get lots of transcription. So what's going on here? So this is a really neat experiment, and what these researchers have shown is that if this is the KLK3 enhancer, well, the KLK3 enhancer is transcribed by RNA polymerase II, and that makes an enhancer RNA, and that enhancer RNA is important for function, for the function of the enhancer. If we eliminate that enhancer RNA, or reduce its levels at least, with siRNAs, the enhancer no longer functions, or does not function as well as it does in the absence of siRNAs. So what that is telling us is that enhancer RNAs are important for enhancer function. Now this is an important finding, and it's a relatively new finding. And there are thought to be tens of thousands of enhancers in the human genome. And we don't know if they all make enhancer RNAs. And if they do all make enhancer RNAs, we don't know if the enhancer RNAs are always important for enhancer function. So we don't know that yet. But what we do know is in the case of the KLK3 enhancer, that its enhancer RNAs are important for its function. And we're going to see